The next topic in this video that I am going to talk about is Clathrin-coated vesicles, its formation, docking, and fusion. First, let me introduce you to some organelles, which are different compartments inside of the cell. Here we have the endoplasmic reticulum, or in short the ER, and the Golgi apparatus. These are all membrane-bound organelles, in which they have the same lipid bilayer structure as the cell membrane. On the right side here, I drew the mitochondria, which indirectly takes part in this process because it is the powerhouse of the cell. The energy required for things to get done in the cell comes from here. So why is Clathrin-coated vesicle important? It is because these vesicles play an important role in intracellular transport, or the transport inside of the cell. You see, the functional machines of the cell are proteins. After they are built and modified completely, they need to be delivered to the right factory for it to function properly. There are also other biomolecules that just can't deliver themselves to the right place in the cell, therefore we need vesicles. They are the cell's cargo delivery men, and this cargo can be delivered in two pathways, secretory or endocytic pathway. The secretory pathway is generally described to deliver proteins from the ER to the Golgi and out of the cell. In the opposite direction, things from the external environment enter the cell through vesicles and are delivered to lysosomes for digestion. I wanted to talk about both pathways because both of them required Clathrin-coated vesicles, our highlight at the moment. Regardless of the pathway that the vesicles take on to deliver the cargo, how these Clathrin-coated vesicles form and successfully land on the target destination and deliver the cargo is the same. Therefore, I am going to dissect this process and use the secretory pathway as an example, specifically the path where cargo is delivered from the ER to the Golgi. So here we are at the membrane of the ER. The inside of the ER is called the ER lumen, and the internal environment of the cell is the cytosol. Inside of the ER, we have some proteins that are already modified and ready to be delivered. On the ER membrane, there are some transmembrane proteins called cargo receptors that can bind to these proteins, which are cargoes. The ER knows that it's time to deliver these proteins when protein adaptants come and gather the receptors as they bind to the receptors' tails. They are also gather RAP and V snare proteins to come and bind to the pre-made vesicles. Adaptants also recruit Clathrin proteins to come and form a Clathrin-coated pit that looks like a basket, beginning to shape into a sphere. The last step in the formation of a Clathrin-coated vesicle requires the final touch of dynamin, small proteins that come in and form a ring around the neck of the vesicle and start to pinch off the ER membrane. The vesicle is snapped off and completely separated itself from the ER membrane, fully covered in Clathrin proteins. Its next destination is the Golgi apparatus. Embedded in the membrane of the Golgi are two proteins, T-snare proteins and tethering protein. Remember V-snare and RAP protein that adaptants recruit and bind to the vesicle? Yeah, they are like the captain and the docker of a ship full of cargo. When the vesicle comes closer to the Golgi membrane, T-snare protein and tethering protein will recognize them and make interactions. The RAP protein serves as the vesicle membrane identifier, which is recognized and binded to the tethering protein located on the target membrane, initiating the docking process. V-snare protein located on the vesicle membrane will intertwine with T-snare protein to stabilize the docking process. When these two proteins interact, V-snare and T-snare twist even further to bring the vesicles closer to the Golgi membrane, causing Clathrin coat to start shredding away, resulting in physical contact between the two membranes. As the proteins pull the two membranes closer and closer together with strong forces, they begin to fuse and result in one continuous surface. This is possible because they have the same structure of lipid bilayer, and the lipid bilayer is fluid, remember? So now the change in pH between the inside of the Golgi and the outside of the cell causes the cargo receptor to release from the cargo and drop off the package.